All right, so here is our notes. We're going to do our notes on cell division. And so if you don't have your Cornell Notes template open, please open that now. So you will go to your classroom, biology, and go ahead and scroll down, and you're going to find your notes here under cell division notes. So if you click this open, so I kind of closed it. If you click it open, then what you will see is you will have the cell division notes here attached. You should have yours with your name on it. Go ahead and open that up, and then you're going to type your notes on there. You can also, if you wish, go ahead and write them all down on a piece of paper and then type them in later. But you need to have this to be turned in. And this, once it's completed, you will create a link to your completed notes on page 14 of your notebook, and I will show you in class how to do that. All right, so let's go ahead and let's get started. So we're going to start, and we're going to really focus in on meiosis. I know you've probably been through in 7th and 8th grade mitosis and meiosis, but here in this biology, I'll tell you the difference between mitosis and meiosis, but really we're going to focus in mostly on meiosis itself. Okay. All right, so our essential question today, which you will type into your Cornell notes, are what are the steps to meiosis and what is it used for? Okay, there's a couple key points that we're going to have as we go through our notes. The first thing is prior to meiosis 1, each chromosome is replicated. And so we'll definitely talk about that a little more. We're going to go more in depth in that after we kind of get through some stuff to understand our genetics. So we're going to get through protein synthesis, but we're also going to talk a lot about replication and what are the things that are needed for replication. The second thing we'll go through here in our key points is the cell begins to divide in a way that is similar to mitosis. So the beginning is very similar to mitosis, but you'll notice in the end it's very different. Meiosis involves two distinct divisions. So it has two divisions. The first one is called meiosis 1, and the second one is called meiosis 2. I know, super inventive names. Okay, so here's a little kind of start of an introduction to meiosis here. So sex cells are gametes, which are examples are sperm and eggs in humans, and in most mammals, that is what we call it, must be made by a special type of division called meiosis. So mitosis does not happen in our sperm and eggs for reproduction, only meiosis, okay? Meiosis is a double division process, which we just talked about. It has two separate divisions, which we'll go over here. And another really kind of important thing is the chromosomes are reduced half the original number. So your sperm or your egg is going to have half the chromosomes that you actually have. And we'll find out, we'll talk more about why that is important as well. And then, for example, in humans, it goes from 46 chromosomes, and each of the sperm or egg actually has only 23. Okay. Meiosis has one DNA chromosome duplication, means it only replicates or duplicates our DNA once, and then 2 division cycles. Okay. Chromosome numbers. So what happens to our chromosome numbers we just talked about? During meiosis, haploid, which I really remember that by half. Haploid gamete cells N are produced from diploid. Di means 2 cells, which are 2N, and we'll go over this more. So basically, every cell in your body, your skin cells, your organ cells, your mouth cells, your tongue cells, your teeth cells, your hair cells, all of those are actually diploid cells. They have 2 sets of chromosomes, and so we call that 2N. But in our gametes, our sperm, our eggs are actually haploid, so they're half. So remember, we just talked about how in humans it goes from 46 to 23. All those diploid cells, those have 46 chromosomes, but our haploid are half, and so it has 23 chromosomes. And we'll talk about why that is really important that that actually happens. A cell with the original number of chromosomes is called diploid, or you may hear me refer to 2N. So diploid, know that it's 46 chromosomes. Sex cells, sperm and egg, gametes, there's another word I will use for them, that contain half the number of chromosomes are called haploid. And then to create gametes, a process must divide the diploid number of chromosomes in half. So you will not have a sperm or egg with 46 chromosomes. If that actually happens, we'll talk about how that can cause all kinds of genetic diseases. And it does happen, not for all. I don't think there's ever been a case where it's all 46 or all 23 chromosomes are still diploid. But there are some that don't divide like they're supposed to, and we'll talk about what those are. Why is reducing the number of chromosomes important? So why would it be important that we wouldn't have 46 chromosomes in our gametes? Well, this is why, and we can talk a little bit more about it. But what happens if there are more than 43 chromosomes? Too much of a good thing, okay? So there's too much of a good thing. And then too many chromosomes can result in genetic abnormalities, okay? Abnormalities. For example, the first one you can have is called Down syndrome, and that's actually 3 copies of the chromosome 21. So when it goes through the duplication, which we'll talk about in just a minute, what happens is when those are supposed to duplicate and then separate, the separation quite doesn't happen there with the chromosome 21. So the baby that is born gets 2 chromosome 21s, usually from the mom, and then 1 copy of chromosome 21 from the dad. So instead of just having 2 copies of chromosome 21, they actually end up with 3. And that is what we usually see as what we call Down syndrome. Okay. Number 2, one that's very common, it's definitely less common than Down syndrome, is what we call Edwards syndrome, which is actually 3 copies of chromosome 18. And actually, I had a sister-in-law who had a baby that actually had this. They tested that. They could tell that in the womb before the baby was born. And this can cause all kinds of issues. And so the organs were growing outside the body of the baby. They knew that when the baby was born, probably wouldn't survive very long, and that's exactly what happened. So chromosome 18 can cause a whole lot more issues than that chromosome 21, and that's why you see it a whole lot less. And the third and the least common one that we see is called Patau syndrome, which is 3 copies of chromosome 13. And I say we don't see this one very often, just because these babies usually die inside the mom in uterus, and we don't really see these babies come out or be born alive most of the time. So here's a little picture. So the parents of a first generation, this is why we have to go from haploid or from diploid to haploid. What if, let's say it didn't happen? This could be really wrong. If the mom had 46, the dad had 46, and if we did not go to haploid, then the child would have 92. And then if we kept going on with the second generation, well, now we have 2 parents with 92, and now the kid has 184 chromosomes. So we'd have every generation would double their chromosomes. Well, we can't have that, because as you can see, when we have 3 copies of even just 1 chromosome, not all 23 of them, then what happens is we have genetic issues. So that's why we do have to have this division. That's why meiosis really does have to happen. And you'll also see as we get into genetics, there's another part of meiosis that is very important as well. Okay, so this is a picture of 46 chromosomes. I actually used to do this with my mice. I used to produce this. I used to look at these so I could actually, what we call genotype the animals, or tell what their genetics are. So this is a picture, it's very blown up, of the different chromosomes that are lined up. Okay, so we also have some sex chromosomes down here, X and Y. Of course, this I can tell is of a young male. Maybe I can't tell young, but of a male because it's X and Y. And so when you have the X and the Y tells you that it is male. If it was an X and an X, that would be a female. Okay. And so human chromosomes after duplication. So this is when we talk about in just a minute about duplication, this is what they look like. So you just saw here is what yours may look like, or a man's may look like before they go through meiosis, before anything else. After the first round of meiosis, then we get duplication. So you can see we have chromosome 1, but now we have 4 copies. And that's because now they're replicated. And so that is what happens in that first stage. And these are called homologous chromosome. And so one of the important things to help you is HOMO means the same. So homologous means the same. So these are exact copies. These ones that are kind of tied at the center, it looks like they're tied with a knot, are the same. And we call them homologous because they are exactly the same. Okay. And human has 46 chromosomes. We have 23 copies of homologous chromosomes. Exception is X and Y look very different. So X and Y look super, super different than the rest of them. Okay. And so these are what we call homologous. So humans, again, are diploid. They are 2N. Somatic body cells, we just talked about, have 2 copies of each chromosome. And so in humans, we call it 2N, or 46. And human gametes are haploid, or having only 1 set of chromosomes. N, so 1 copy. So gametes are haploid and have 1 copy, which are 23 chromosomes. And then somatic cells actually have 46 chromosomes, basically 2 copies of each chromosome. Okay. So in humans, 22 of the chromosomes contain the information for our traits, hair color, eye color, etc. And then that 1 chromosome we just talked about is our sex chromosome. That's all that codes for. And so XX is female and XY is male. So this is what we call a karyotype, which I just showed you. So this is actually a female. So I just showed you a male. This would be a female. And the reason I know is there's no copy of a Y. There's actually 2 X's, so this is a female. And you can see the dark and the light regions, and when they actually do DNA analysis that you may hear from the police, actually that's when they start actually mapping out each of these chromosomes. And I used to do that when I worked up at Stanford. Okay. So let's go through the actual steps in meiosis, okay? You're not going to be asked the names and the specific things, but moreover, the general area. But I really want to introduce you guys to the names, so when I'm saying them, you guys can kind of understand them. So step 1 is what we're going to call the interphase, or what we call the interphase. Step 2 is the prophase 1. Remember, there's 2 divisions, so we're going to talk about the first one. So you're going to have interphase, then prophase 1, metaphase 1, anaphase 1, telophase 1, and then steps 6 through 9, like I just told you, there's 2 divisions. We do not do a duplication, which we'll talk about what happens in interphase, but then we're going to go back through steps 2 through 5, okay? All right, so let's talk about step 1. So this is interphase. Remember when we looked at the cells? So this is our nucleus in here, which contains our DNA. So it's the same as in mitosis. So this is a step that exactly is the same in mitosis and meiosis. And the one thing I tell between the difference is mitosis has a T in it, so it makes 2 cells. And I'll show you how that kind of helps me remember which one is which as we get to the end. And then cells undergo a round of DNA replication. So that is what happens in interphase. It's really the only thing, okay? And then forming duplicate chromosomes called homologous. So you remember that I showed you those 2 copies are the same chromosomes. So that is what happens in interphase. So then we get to step 2, and now we have those ones that are tied together a little bit. Remember our homologous. So our homologous are shown here by color, so purple and green. And so this is what happens in prophase 1. So this is the start of what we call meiosis 1. That interphase is kind of the precursor, our beginning, before we actually start meiosis. And so what happens here in meiosis 1? Each chromosome pairs with its corresponding homologous chromosome to form what we call a tetrad. And we call it a tetrad because tetra means 4. And you can see it has 4 kind of legs. 1, 2, 3, 4. So this purple one, that is a tetrad, and the green one is a tetrad. And these two purples are homologous, and these two green are homologous, okay? But a very unique event, and it's very, very important for why you say if you have siblings, they do not look exactly like you. If they have the same mom and dad just as you do, there's a reason why they don't look exactly like you. And here it is, because this is what happens, what we call crossing over. And it happens in prophase 1. So what happens, homologous chromosomes pair up and form tetrads, then exchange portions, and each single one is called a chromatid, and it results in new combinations. So basically what happens is the legs cross over, and so a little bit of this purple will switch with the green. And so what will happen eventually here, so you can see right here, sister chromatids, is you will have a switch. So they will actually switch over and they will move from one to the other. And that's what we call crossing over. So a piece will come over. So you can see here we've got our tetrad, blue and red. We see they start crossing over. Things happen. And now a little bit of the blue is on the red and a little bit of red is on the blue. And that's what causes that whole variation, why, you know, even in my kids, my girls have brown hair, my boys have blonde hair. All this crossing over causes all the kind of genetic variation that we see. And so it's very important because we don't need everybody to look like everybody else, okay? All right, so here's another example with maybe some letters. So you can see they cross over and touch, and now there's a little B here and a big B over here. So it just happened. Why does it happen? We do think it causes, you know, it's one of those things that happens in nature that actually makes things better. We just know that this is why it happens, okay? All right, so let's talk about step 3 now. So in step 3 is metaphase 1. And we start seeing the spindle fibers, what we call the spindle fibers, show up. Everything kind of lines up down the middle, and things are starting to be pulled apart. Remember, we have our homologous chromosomes, but this is 2 copies of them. We don't need 2 copies in our gametes, so now we actually need to split them apart, okay? And so that's what happens, in metaphase, is they start showing up to actually separate these homologous pairs, okay? And then step 4 and step 5 is what we call anaphase and telophase. So what does that mean? What happens now is we're splitting up the homologous pairs with our crossing over, right? In anaphase 1, and they're pulled to the separate sides of the cell. In anaphase, so you can see they're starting to go towards their side. And then telophase is when we actually get the center part here to show up. They start creating the nucleus again around it. Here you see. And then cytokinesis is actually where it starts to actually cut down the middle, okay? And so in cytokinesis means to separate. So that's when they actually completely separate into 2 cells, okay? And then we go into what we call mitosis 6 through 9. And again, we go through prophase 1. We have our crossing over, okay? And the nuclear membrane disappears. We have our crossing over, and then we head into metaphase 2 here, where they line up. They're going to get split apart, okay? And then in anaphase again, they get separated, pulled to the sides. And again, what's going to happen again is each number will have half, and then we'll go through our telophase and cytokinesis too. So the same thing happens. Everything happens again, just so that, again, we can now have our haploid cells, our N, okay? So in meiosis, in the end, we actually, from one original cell, will end up with 4 non-identical, what we call daughter gametes, are formed with half the chromosomes. So we will not get identical cells. They will all look different, and we will actually get 4 of them, okay? So here's a little picture of what happens. So we have prophase 1. We have our crossing over. Things start lining up. They get separated, and then we get telophase and cytokinesis, where we get 2 cells, but we still have our sister chromatids together. Then we go through meiosis 2. Remember, we only went through the duplication before meiosis 1. It will not duplicate. So now we're going to go to our prophase again. We might have some crossing over again. And then we have our metaphase. They line up. Now we're going to separate them, the ones that are tied together. We're going to separate them to actually end up with 4 non-identical cells. Okay, so this is the big kind of idea between mitosis and meiosis. In mitosis, which happens in your body all the time to repair your cells, is you will start with your 2N, 46 chromosomes, and you finish. So basically you go through a duplication, and then it comes through and it makes 2 identical cells. So in mitosis, you make 2, and there's a T in there, identical cells. When you go through meiosis, you will not get 2 identical cells. You actually get 4 non-identical cells because we go through 2 divisions. So we start with 2N and finish at N. Meiosis only happens in the gametes. Mitosis happens in the rest of the body. This is what happens as a baby grows. Meiosis only happens in the gametes, okay? All right, so those are our notes for right now. You can go ahead and finish those up and figure out, oh my God. So you want to make sure at the end that you process your notes. And so what does that mean when you process your notes? You're going to go back, you're going to add questions or connections to your notes. You have in the left column, remember when we went through elementary school and you talked about connections, you made personal connections, prior knowledge, anything you may have or remember in your brain that you remember about division, mitosis, or meiosis. You want to add those, and then you're going to use those connections and questions to create a 3 to 5 sentence summary at the end of your notes. And then also make sure you create a link in your notebook to your notes and turn those in. All right, if you have any questions, please let me know.